Hi, my name is Jeremy Comel, currently a postdoctoral researcher in Crystal Pollitt's lab at Yale University. All the software that I'll discuss here and all the open source software we've designed over the last 5 years can be found at innovative-omics.com. This work is a collaborative effort with Agilent Technologies and University of Florida Southeast Center for Integrated Metabolomics. We are in the midst of the omics revolution. In this revolution, instead of measuring a few compounds based on a specific targeted question, we are now able to understand an entire system by measuring thousands of molecules across different classes, whether it be lipids, small molecules, contaminants, proteins, or genes. This revolution is possible because of mass spectrometry, specifically high-resolution mass spectrometry, whereas tandem mass spectrometry provides unique and universal information that can give structural information on any type of compound that's ionized. Therefore, using high-resolution tandem mass spectrometry, we've generated a number of libraries, both for lipidomics, and now we've extended our algorithms to PFAS, a contaminant of concern. The problem is, even if we have these libraries, we still need to select ions to fragment in order to match against the libraries. And if these ions aren't selected, we will miss important identifications. In most omics studies, we combine mass spectrometry with a separation technique, such as in this case, liquid chromatography, and this is a lipidomics dataset for plasma. Now, as the compounds elute across time and then ionize, we're racing to select the most important ions and fragment them. For example, if we look at the triglyceride region in this time mass to charge plot, in this 15,000 millisecond region, if our scan rate is 1 scan per 250 milliseconds, we can only obtain 60 ions here, which we can fragment. Problem is, there's 400 features in this region, so we can only get fragmentation for 1 out of 8 compounds. You can imagine that as this little girl reaching through this box and pulling 1 out of 8 and hoping that it's the compound of interest, whether it be a biomarker or lead to new discoveries on a disease mechanism. Traditionally, what is done to select these ions is at a specific time, a full scan is taken, and in this full scan, the top 5 most abundant, or top N, you can choose your N, are selected and fragmented. And so in the end, we only get fragmentation for the most abundant ions. In biology, it's the least abundant ions that are often of interest. For example, those that are drug targets, that are bioactive at very low concentrations, or those that are signaling molecules that are very important to understand disease etiology. These will all be the low abundant compounds, whereas the high abundant ones often are structural compounds such as phosphatidylcholines, which might not give any information related to the questions we have. Therefore, in 2017, we released an application of iterative exclusion and a script in order to exclude the top 5 most abundant ions, or top N, and generate an exclusion list. You re-inject the sample, that exclusion list is applied, and you acquire the data for the next top 5, and then the next top 5. And therefore, sequentially, you can get coverage of all ions in your experiment. Here's the actual data for that plasma lipidome we were looking at earlier, where each dot is an ion that's selected for fragmentation. On the right, you can see as we have new acquisitions and apply exclusion lists from the previous run, we get more and more coverage of ions that are selected for fragmentation. So here you can see, or barely see, that the dots in gray were the ions that would be selected just using data-dependent analysis alone, whereas all the colors show the new ions that are selected for fragmentation. And therefore, we can see that we get a new rich diversity of ions selected that weren't selected previously, and this carries over to the number of identifications as we'd expect. So in data-dependent analysis alone, as you acquire 6 samples, let's say this pooled plasma, just by stochastic nature of the technique, you get some increase in identification. But when we apply iterative exclusion, we nearly double the number of identifications in this case. Now, we don't only just double the number of identifications, those identifications often express unique biology that wouldn't be covered otherwise. So, for example, on the left, as you'd expect, the first injection, the ions selected are high abundance, whereas at the sixth, they're orders of magnitude lower abundance, those ions selected for fragmentation. And those are the most common diglycerides, for example, if we look at one class of compound identified in the first injection, those that have even chain fatty acids and are produced endogenously. But by the sixth injection, we get short chain and odd chain fatty acids within the diglyceride, which relates to lipids from the microbiome and produced at trace levels endogenously by other biological mechanisms. And so we get information that might be very important for our research that we would have missed otherwise. But you may ask, if you're familiar with mass spectrometry, what about data-independent analysis? What about all-ion fragmentation? What about SWAP? What about sonar? What about all the other techniques you can do where you can select, or instead of selecting ions, you fragment everything simultaneously? So, in a data-dependent fragmentation, you take one ion, you fragment it, and you look at those fragments. And if you look on the right, that means that within a long cycle time, for example, of 4 scans, you only acquire a small portion of the mass to charge region. Whereas in all ion fragmentation, for example, or all ions, you can fragment everything simultaneously because you're not selecting one by one. And therefore, in only 2 scans, you can have fragmentation across the entire mass to charge region. The problem is, in data dependent, it can be relatively simple to reconstruct the structure because you have all the pieces, and if you have an MSMS library, you have the instruction manual, you can build that Lego set. But in data independent analysis, now maybe your hobby is putting together Legos, maybe your child's hobby is taking apart Legos and throwing them all over the rug. And so it takes all your Lego sets, puts them on the rug, and now you don't know which Lego piece belongs to which set, and it's a headache trying to build back what these structures are. So what we have to do is take that precursor ion and correlate it in time to fragments to make sure that those fragments come from that precursor. The problem is, for example, this fatty acid could exist in many different lipids, and so it's hard to deconvolve. And then there's other fatty acids, for example, this one that's even more common. And so our deconvolution algorithm might actually not associate this fragment with this lipid, and we therefore get a false negative. We don't identify it. And that's why even just using traditional data-dependent analysis and our algorithm for deconvolving all ions, at least in lipidomics where it's very complex, data-dependent analysis still does better. So ion selection is still king. Using iterative exclusion data-dependent analysis, we almost cover all of the ions from all ions, as well as many, many more, showing again this advantage. We published this work in 2017, and it has over 50 citations, and many people are using this in fields ranging from lipidomics, metabolomics, contaminants, but all the way to addicomics, and it's very interesting to see the unique applications. The problem is that technicians and researchers don't like the technique because of how tedious it is. So this is how a researcher, once they see this wonderful talk, what they would actually do. They'd come to their instrument, wait for it to acquire data. They bring it, run the IE-omics script, generate an exclusion list, bring it back to their instrument, upload the exclusion list, rerun the instrument, bring it back to the computer, rerun the software, bring it back to the instrument, and so on and so forth, acquisition after acquisition. They could spend an entire day just doing this process, which is very tedious. Therefore, it hasn't been applied as widely as we'd hope. Some of the instrument companies are now taking this work on. So for example, Agilent has a software very similar to IE-omics, pretty much does the same thing, whereas Thermo, we know, have IDX, and other vendors are also creating their own iterative acquisition approaches. And so that can be advantageous, but many instruments still don't have these techniques. And researchers, it would be very beneficial if we had an open source platform we could continue to improve that works on all vendors. Therefore, the one technique that I can think of that would work well in this case is that we take a single scan, and from that single MS scan or set of scans that we run all at once, we then predict and decide all our fragmentation sequences just based on those scans. So for example, in intelligent iterative exclusion is what we're terming it, you would take the top and most intense peaks from a full scan data file, combine all the ions across retention time, and generate an exclusion list. And then you'd repeat that process, and then the next time you take the next top most abundant ions, append it to the original exclusion, generating your second exclusion list for your second injection, then your third for your third, then your fourth for your fourth injection, so on and so forth. And then you can upload all these simultaneously and let the instrument run instead of waiting for each acquisition. In the iterative inclusion or intelligent iterative inclusion process, or III, now we're looking just for those ions that would be of most interest to get fragmentation from. So it's a slightly different philosophy. So in this case, we're using XCMS for peak picking, and therefore we're reducing the ions we select only to those that form nice peaks. Then we're using blank filtering, which is going to remove any ions for selection for fragmentation that are just from background noise, such as from the instrument or solvent, etc. Then we do MS1 level annotation, which is optional, where you can take whatever your MSMS library is that you're going to search against and only look and see if those features have exact masses that would be in your database in the first place, so you don't waste your time selecting ions that you have no database to match against anyway. And then adduct filtering, so for example, maybe you don't want to select and fragment sodium ions, or maybe you have a solvent system with formate, so you don't want acetate ions, and so this can reduce your list further. Then the software generates a peak table, and from that peak table, you can either create a single inclusion list, but let's say you have 100,000 features on that peak table. If you were to put it on one inclusion list, the instrument would try to compete between those different ions at a specific time and only acquire data for certain ions on that inclusion list. So then we can do multiple injections, and the software will split the inclusion lists into multiple inclusion lists, for example, by retention time, so that the inclusion lists are evenly spaced out across time to get the most scans across peaks. The advantage of III is that you have less injections. Often, even a single inclusion list might be generated, just one single injection to get all the ions of interest. There's deconvolution of isomers that's possible because now that you have an inclusion list, you can use a targeted approach and get 10 or more scans across your peaks or closely eluding peaks and then deconvolve them by MSMS. And it's possible to include information for every sample. You could take all of your MS1 data and make sure that even if an ion only appears in a few samples, that you still select and fragment it. Now, the disadvantage is that it's semi-targeted, and so things that, for example, don't form a nice peak won't be included for ion selection. And therefore, you can combine this with IIE, which is more comprehensive, so that you have coverage of everything in case you want to look back or your databases expand, etc. Now, our lab, we haven't tested the software except for computationally yet, and so the next step is for you guys to start taking this software that's fully developed and we have instruction manuals and start applying it and seeing how well it works. But based on applying it to old data, we see that just using a single injection of III will get the coverage of iterative exclusion, data-independent analysis, and data-dependent analysis within the second injection, just in a single injection. So very comprehensive in a single injection. And then if you look on your right, we can actually see the process for this PFAS data set in this case, where XCMS pulled out 16,000 peaks. Blank filtering, we get 14,000 peaks. And then using identification based on our database, there's only 207 ions that we're going to select for fragmentation that we can match up against our database. So in conclusion, we provide a free software which increases molecular MSMS coverage based on a single injection. And now we need testers. So please download the software and contact us about your applications.